My name is Norma Andrews. I'm a professor at Yale University, and what I'm going to be talking to you about in this second part of my lecture is about Leishmania, this parasite, and the disease that it causes in humans, which is called Leishmaniasis. So this is also a protozoan parasite from the order Kinetoplastida, family Trypanosomatidae, and inside this family we find several protozoan organisms, and the two groups that are medically important because they cause serious diseases in man. Trypanosoma, which includes Trypanosoma cruzi that I already talked about in the first part of this lecture, and also the African trypanosome that causes sleeping sickness in Africa. Leishmania, which I'm going to introduce to you now, is closely related to trypanosomes, and it's also an intracellular parasite. So here in the distribution of Leishmania throughout the world, we can see that it's not only restricted to South America like Chagas disease, but is also found in many regions of Africa, the Middle East, and also around the Mediterranean. So similar to Chagas disease, Leishmaniasis is also largely a disease of poverty. It is mostly found in underdeveloped regions of the world, but it has some characteristics that actually make it very serious in the sense that development is proving not to be sufficient to stop transmission. And actually there's several signs that the disease is recently moving from rural areas to urban areas of the world. So this is a very serious development that indicates that this number of people, which are currently around 12 million that are already infected by this parasite, there are 300 million people at risk worldwide. So the incidence of Leishmaniasis may actually increase over time. So here we see an example of one of the clinical manifestations of Leishmaniasis. This is a child with cutaneous lesion, which the form of the disease in Leishmaniasis is directly associated to the species of Leishmania that causes the infection. So these three species here, L. major, L. tropica, and L. mexicana, are frequently associated with the cutaneous form of the disease, which can be very disfiguring, although these lesions, most of the time, they are self-healing. There's a more serious form of this cutaneous disease, which is the form that attacks the mucosal membranes, in which this can really be seriously disfiguring, and this is usually associated with some of the species, for example, Leishmania brasiliensis. The most severe form of Leishmaniasis is by far the visceral form, which is caused by these three species mostly, Leishmania donovani, Leishmania infantum, and Leishmania chagasi. And this disease is characterized by periodic fever, severe enlargement of the liver and the spleen, and also weight loss and anemia. So this is a significant cause of morbidity and mortality in the endemic areas for visceral Leishmaniasis. This disease was already known to be present. It was called early on Kala-Azar, but what was not known is what was the organism causing it. It was not even clear that it was an infectious disease. So it was this British physician, William Leishman, which analyzing material from patients in India, and also simultaneously by the Irish investigator Charles Donovan, they realized that macrophages present in the spleen of these patients were heavily loaded with these organisms, which they recognized as similar to trypanosomes. So this was the first indication that this was a parasitic disease. And another very important finding in the history of Leishmaniasis was made by Rogers and Nicole a few years later, in which they took this material from the spleen of the patients and put into culture. And they were able to show that a second form of the parasite appeared in these cultures, indicating directly the existence of a life cycle and most likely an insect vector. So this actually was only decades later that the first hint for which could be the insect involved in this transmission was made by John Sinton when he realized that the incidence of visceral leishmaniasis in eastern India actually coincided with the distribution of a specific type of sandfly, the silvery sandfly. And this rapidly then led to a lot of work in which this picture is what emerged, in which these regions that I already showed that we can see Leishmania being transmitted to humans. In each case, a specific species of Leishmania is associated with a specific species of the sandfly, which is responsible for the transmission. And it was only more recently, decades later, after these original observations, that there was a formal demonstration that sandflies could transmit Leishmania to humans, although earlier it had already been detected in sandflies that had been feeding on over here in an infected animal, but also in humans. So it was then only at this time around the 40s that the cell cycle, the life cycle of Leishmania was really understood, with this replication of the parasites happening inside the sandfly. This is similar to Trypanosoma cruzi in the sense that these parasites also don't gain access to the salivary gland. They are transmitted in this case by regurgitation. So after a blood meal, this fly delivers these parasites, which replicate abundantly in its digestive tract, and this is how they penetrate the mammalian host, which in addition to humans, dogs and rodents play a major role in this cycle, and it is inside macrophages of the mammalian host that the intracellular replication occurs. So more recently, David Sachs at the NIH made another very important observation and contribution to the understanding of the biology of Leishmania parasites, which was by identifying an infective stage. So similar to what I discussed in the first part of this lecture for Trypanosoma cruzi, Leishmania also has an infective stage. So the parasites that replicate inside the insect vector, they replicate into these infective stages, which David Sachs called metacyclic promastigotes. And during this differentiation, they also acquire that resistance to lysis by complement, which allows them to survive inside the mammalian host. So David Sachs and other groups have made a very important contribution in linking this differentiation into the metacyclic form to this surface molecule LPG, which is a major component of the surface coat of these parasites. So what is shown here in this immunogold electron microscopy, these black dots that we can see here in this enlarged segment represent antibodies reacting with this surface molecule. And what David Sachs and others showed is that these molecules, which are lipid anchored, and they have these repeating disaccharide phosphate units, this molecule undergoes a significant elongation during differentiation, and these side groups, which are usually terminated in galactose in the epimastigote insect form, acquire these side chains terminated by arabinose. And this is a key change in one of the specific species, Leishmania major, that David Sachs showed that is responsible for a very important part of this transmission cycle, which is the detachment of the parasites from the midgut of the insect, allowing them to be delivered to the infected host. So this change in the structure of this LPG molecule is what mediates detachment and then facilitates transmission during or after the blood meal by the sandfly. Another very important point that emerged recently also from the work of David Sachs at the NIH is that the vectorial competence, the ability of these sandflies to transmit specific species of Leishmania, is related to polymorphisms on this LPG molecule. So only now, very recently, that a complete and much more complete picture has emerged of what are the molecular properties of this parasite that really create these very specific associations between vector and cycles of transmission throughout the world. What is the next frontier now? Now that we really understand quite well what is happening inside the sandfly with delivery of these infective stages, the next frontier is to understand what's happening in the mammalian host. So we know that Leishmania mostly replicates inside macrophages since the original observations of Leishman and Donovan, but we don't understand completely how the parasites actually survive in these cells. So macrophages are cells that are actually designed to destroy intracellular pathogens. And the entry of Leishmania into these macrophages is known to be mediated by phagocytic receptors. So in this case, different from Trypanosoma cruzi, there's no indication of an active mechanism of invasion, but it is pretty clear that at least the complement receptors, CR1 and CR3, play an important role because these parasites are not lysed by complement, but they are opsonized by complement, which allows them to be efficiently internalized by macrophages. Inside the macrophages, the really remarkable aspect of the life cycle of Leishmania is that these parasites can survive in compartments that have many properties of lysosomes. So they are acidic, they contain a full set of hydrolases and also of lysosomal membrane proteins. And we don't really understand how the parasites are able to spend their whole cycle, which also lasts several days. They have a doubling time of around 12 hours. And what is indicated in this slide here is that even MHC class II, which is a protein expressed by activated macrophages, it is present on the membrane of these vacuoles containing the parasites that replicate very well in spite of these harsh conditions. And so in addition to this remarkable adaptation to survive inside cells of the immune system, which in normal circumstances actually play a role in the elimination of pathogens, another very challenging aspect of Leishmaniasis is the difficulty in limiting exposure of humans to these insect vectors. So in this case, this is a very small insect. What this picture here shows is one sandfly sitting on the knuckle of a finger, just to give an idea of how small they are. So the simple measures that have been shown to be so effective in containing malaria transmission, for example, just bed nets, really are not effective because these are very small flies that can go through these regular bed nets that are normally used in these endemic areas. So what is very clear for Leishmaniasis is that there is a strong need for development of new drugs, and new drugs with less toxicity, because again here the drugs currently in use in large parts of the world are still very toxic. The good news is that recent developments are leading rapidly to the identification and implementation in clinical trials of new and better drugs, with several philanthropic organizations and also non-profit institutions participating actively in this concerted effort to make this very important need in public health met by taking advantage of current state-of-the-art technology. Thank you. In the next segment, I'm going to discuss more the cell biology of infection of both Trypanosoma cruzi and Leishmania.